It's my pleasure to introduce our first speaker, who is Melanie Boly, MD, PhD. She's a neurologist, a medical doctor. She's worked with really all the big names in coma research, Adrian Owen, Stephen Lories, now Giulio Tononi. She's technically currently the postdoc researcher at the Belgian National Fund of Scientific Research and neurologist, well, you're finished training now. And as I said, she is currently working with Giulio Tononi in Wisconsin. She's done a number of very interesting and important works in detecting awareness during coma and the neurobiology of coma. So please welcome Melanie Boly.
Thank you very much. It's working. First, I want to thank the organizer for inviting me. It's a great pleasure to be here. So what I'll do, actually, Stephen Lories is also talking tomorrow, and as we work 10 years together, there's some parts of work we share. So I won't present everything. The focus of my talk today will be some kind of general introduction to neural quality of consciousness in vegetative states and doing also some parallels with sleep and anesthesia. So I'm a neurologist in training, a clinician. And as clinicians, we define consciousness, so clinically, by two main components. The first one is wakefulness, defined by eye opening, and the other one is awareness per se. And usually these two components go together, they're correlated. So for example, during sleep, when your wakefulness decreases, when your arousal decreases, your awareness will decrease too. Same phenomenon during anesthesia or coma. Vegetative state is a particular clinical entity because the patients have the eyes open but show no signs of awareness. Actually, vegetative state is not the only state where there is a dissociation like that. You can find such a dissociation also during somnambulism or in some seizures. Minimally conscious state is defined by, actually, they are very difficult to differentiate these patients from vegetative state patients at the bedside, but they are defined by the presence of non-reflexive behaviors. So for example, a response to command or some emotional reactions to stimuli. So these patients show non-reflexive or purposeful behavior, but they are unable to communicate. And then locked-in syndrome is not a disorder of consciousness, but these patients are fully aware, but unable to communicate their consciousness. So they are part of the differential diagnosis, but they are fully conscious. The problem we have with these patients is actually diagnosis at the bedside is very difficult. And if you don't use the appropriate scale, you have as much as 40% of misdiagnosis. So it's very difficult to make the difference between these patients. So in the last few years, and Stephen will talk more about that, there has been some progress in trying to detect consciousness in patients that are clinically vegetative or in minimally conscious state by trying to bypass this clinical examination and use functional neuroimaging. So for example, you ask the patient, imagine playing tennis, and you look if in the functional MRI, there's a brain activation of motor areas, for example. These techniques are really useful. The problem is they work only in a minority of patients. And if you have a negative result, then you're stuck. You cannot say anything about the state of consciousness of the patient. Maybe the patient didn't want to respond. Maybe he doesn't understand language. You have so many reasons why you cannot interpret a negative result in these kind of paradigms as the absence of consciousness. So then you're a bit stuck, and you want maybe still to have some information about general brain function and ability for consciousness in these patients. So if you don't want to use a response to command, then actually you switch to another approach, which is called passive paradigms. And as we say, actually at the individual level, we don't really know if the patient is not responsive clinically, if he's conscious or not. So what we do is obtaining different measures of brain function in these patients, and then comparing them to brain function in other states such as sleep, anesthesia, epilepsy, trying to find actually some general principles that can differentiate when you're aware and when you're unconscious. So at the end of the day, when you use this kind of passive paradigms, you end up trying to find some reliable neural correlates of consciousness and trying to find some generic mechanism for like brain ability to generate conscious perception in not only vegetative state, but the other states. Okay? So that's kind of the approach I will talk about here. And so just something to mention that actually when we use this kind of approach, we are talking about neural correlates of the level of consciousness. So in sleep, anesthesia, and coma. And it's very complementary, but it's kind of a different approach than studying the neural correlates of consciousness in healthy volunteers. The question is different. And there are other differences, but there are two main I want to underline here. So the question here is there any kind of perception? It's like a potential. Is the ability to perceive anything there? While in healthy volunteers, the subjects are aware anyway, but you're looking if they perceive like one particular content consciously or not. It's a kind of different question. And also, importantly, the correlates described in the literature are different in a way that in healthy volunteers, usually when you don't perceive something, the brain activity will be smaller than when you perceive something consciously. And actually, what I will try to show you here is that in disorders of consciousness, the level of activity is actually not sufficient. You also need another criteria, which is the differentiation of brain activity. And so using a paradigm that you use in healthy volunteers to detect consciousness or not to patients is a bit tricky. It's kind of different things. So what I'll talk about here is actually these two dimensions I just cited. So I will show you different kind of connectivity studies and show you that there are two main things that you can find as a difference between a state of full awareness and then when patients are unconscious during anesthesia, coma, sleep. And that's either you have a breakdown of connectivity or you have a loss of differentiation of brain activity. So I'll talk about three different approaches to study connectivity in the brain. And that would be first functional connectivity studies and then affective connectivity. So perturbational approach using transcranial magnetic stimulation or external stimuli. These techniques, again, provide different kind of information and are very complementary. So the first studies we performed on functional connectivity in vegetative state patients were using resting state functional MRI. So just before that, we were looking at metabolism. So using PET scan, for example. And when you look at these metabolic studies, a common finding in patients in vegetative state is that there will be a widespread decrease of metabolism, not in all the brain, but in this network that you see there in red. Yeah? So a widespread frontoparietal network, both on the midline and on the lateral side. And actually, there's increasing evidence that this widespread frontoparietal network can be divided in several entities. So the first one would be on the left, simplifying very much again, some areas, mainly on the midline, which are actually areas of the default network, and people have involved default network in awareness of self, self-referential processing, memory, some like more related to self-perception. And then the areas more on the lateral side, there in red, are actually typically involved in awareness of the environment, so consciously detecting stimuli. So, well, I'll show some studies about processing of external stimuli. That's kind of a way also to study that dimension. But studying default network function in patients in vegetative state that cannot communicate is quite tricky. And actually, so we were very interested in these functional MRI studies where you can actually looking at correlations between the signal in these regions and study the connectivity like that without requiring collaboration of the patients. And so we looked at if this resting state functional MRI connectivity was a good marker of consciousness in patients in vegetative state. And it is. Actually, there is, when you look at functional connectivity in the default network, a very good correlation between the connectivity virtually in all the key nodes of the network and in the thalamus too, and the level of consciousness of patients. So this seems to work indeed. And then it's the same in anesthesia, actually. As I said, we like to compare to anesthesia or sleep to see what's happening. And in anesthesia here in the propofol, you have 4 different conditions. You have wakefulness, sedation, and consciousness recovery. And you see that the more you go deep in sedation, the more your connectivity in the network will decrease. And then it's actually recovering when you recover consciousness. And it's not only true for the default network, but also for the lateral frontoparietal cortices. Same story, thalamocortical connectivity in these frontoparietal networks correlate to consciousness. with consciousness. It's not the case for lower level networks. So in visual or auditory networks, the connectivity is preserved independently of the level of sedation. And even in complete unconsciousness, when you completely lose response to command of subjects under propofol, you will have preserved thalamocortical connectivity in these networks. In sleep, it's a bit different in a way that total brain connectivity is, that's a recent study we performed where we really quantified the integration, the connectivity in the brain. And it doesn't seem that the total brain connectivity really decreases, but there is a reorganization in independent modules. So there's an increased brain modularity. It's a bit different still. There's a breakdown of the general global brain network in some more independent parts. Okay. So indeed, this seems to work. If you look at functional connectivity using resting state fMRI, you have a good correlation between the level of connectivity, the strength of connectivity in frontoparietal cortices, not in lower level cortices, but in frontoparietal cortices, you have a good correlation between the strength of connectivity and the level of consciousness. The thing is, functional MRI, resting state functional MRI, is a signal that we don't really know. There are some kind of debates on the functional significance. That's one thing. Though now it's more accepted to be really linked to neural activity. And the second thing is, when you look only at correlations, you're not really sure that it's a product of direct causal interactions between the areas. If you actually stimulate directly an area and then you look where the signal propagates in the brain, then you're closer to causal interactions in the brain. And that's what is called effective connectivity, or what you can do actually using what we call perturbational approach. One example here is kind of the royal example, is transcranial magnetic stimulation, TMS. So you stimulate a part of the brain, you induce some current there, and then you record with electroencephalography how it propagates. And there you really look at causal connectivity in the brain. So that's kind of a good check to see if indeed connectivity is decreased in these states. And so the first study that was performed actually using TMS-EEG in disorders of consciousness was during sleep by Marcello Massimini and Tononi in 2005. And what they did is, so here you see actually the result of source reconstruction of the brain activity induced by TMS. And you see that in wakefulness it's spreading to all the brain, and then it's complex. Actually, you have, if you look at the source-reconstructed signal of different areas, you will see that the different areas do different things. So the signal is moving across the cortex, and it's like complex and connected. During sleep, actually, the response will be very focal. You have a breakdown of connectivity, and it's stereotypical. And then we wanted to confirm that also during anesthesia or coma. Is it also the case? So we used TMS-EEG in collaboration with them. And there are now a few studies showing that. For example, here is an example of patient in vegetative state, then recovering consciousness. And you see that during vegetative state, the response is much more focal than when the patient recovers. Same story during anesthesia. There's a study in midazolam that is published. We've done also that during propofol and also importantly during ketamine, which is a very different mechanism. And in all these states, you have a breakdown of connectivity assessed by TMS-EEG. So that's pretty good because it's indeed showing to us that there seems to be a good correlation between consciousness and brain connectivity. Well, clinically, the thing is, TMS-EEG is very difficult to do in patients. So we wanted to check also if you can have the same kind of information with another perturbational approach, which is actually using external stimuli. When you use external stimuli, you're also doing some kind of change in the brain, and you can look at causal propagation of the activity. And actually, so that's also like a valid perturbational approach. Though you don't directly target the cortex, you do causal perturbation in brain activity, and then you can look at effective connectivity changes like that. Actually, responses to external stimuli, we use that since quite a while in patients in vegetative state and minimally conscious state. That is, for example, a PET scan study where we look at activation in the brain in response to clicks, to very simple auditory stimuli. And what you can see here in yellow, is the activation in healthy volunteers. You have primary cortex and also associative auditory cortices. In vegetative state patients, at the group level, you have only activation of the primary cortex, but it doesn't spread more. Minimally conscious state patients here in green, they activate more, not only the primary, but also the associative cortices. And if you look at the functional connectivity between the auditory cortex and the rest of the brain, you will see that patients in minimally conscious state, they have much better like the connectivity is preserved between auditory cortices and then some also frontoparietal regions that I said to you we think are important for conscious perception. So that's very encouraging. The only thing is, this is PET scan and this is at the group level. But when you look at in individual patients, the story is more complex. Actually, some individual patients will activate more than the primary cortex. Even in some patients that are vegetative, like here, these two examples here, in response to strong stimuli like the own name, you can have activation of associative auditory cortices. And what we do usually to interpret this kind of data is a framework a bit like Stanislas Dehaene uses for healthy volunteers, saying, okay, if you activate the primary cortex only, it's unlikely you're conscious. If you activate the associative cortices, where it's not so much frontoparietal, so maybe you're not conscious anyway. What you need to be conscious is actually maybe our criteria should be activation of frontoparietal cortices. That could be working. So that's what we thought for a long time, and then there were some new studies coming, and here again is an example in healthy volunteers, but showing that you can activate the frontal cortex subliminally. And then we have now some other examples during anesthesia. Here, for example, it's a volunteer where you present the own name, again, a strong stimulus during wakefulness, and then under clinical loss of consciousness under propofol, no response to command, and no report after of any perception. So in wakefulness, that's the kind of network that is activated for this volunteer. And then here is during propofol. I think it's kind of a good example to show you that, well, even when you're unconscious, you can have some kind of frontoparietal activation. This is not a sufficient criteria to really distinguish at the individual level someone that is conscious or not. So, well, we thought about what could be left, yeah? What could be like the critical bottleneck. And then the hypothesis was, okay, maybe you need activation of the specialist cortices, and then you need the activation of frontoparietal cortices, granted. And actually at the group level, it's really true. You see less activity in frontoparietal cortices. But maybe what is really limited, like what is really the bottleneck, like the common phenomenon to everything, is that not only you need activation to go to frontoparietal cortices, but also you need to go back. So some kind of backward connectivity. So that's what we thought, okay, maybe we'll try to test that. And so actually there are some new techniques now to analyze, for example, evoked potential that were developed by the team of Carl Friston in London. It's called dynamic causal modeling. What you do there is actually create a mathematical model, and you see like a realistic one, biologically informed. And you try to see what kind of neuronal architecture could create the type of signal you observe. And so you can really decompose, for example, for auditory stimuli, you say, okay, I need my primary auditory cortices, the temporal cortices, and then the frontal cortex, and then I can measure connectivity going forward and backward and see if there is a change between conditions or between patients and controls, yeah? And so they've done that in healthy volunteers, and they've done also, in particular, an interesting study in healthy volunteers showing that to explain the short latency evoked potential, so the very short lasting signal you have when you present a sound in the electroencephalogram. Actually, the activity of forward connections were enough. But then, when you look at longer latencies, like several hundreds of milliseconds, then the backward connections are really needed to explain the kind of activity you see. So there seems to be, if you summarize all that, that I just said, that frontoparietal activation granted is important. And when we see that at a group level, it seems to be very important as a general principle, but maybe not sufficient as a criteria to distinguish consciousness at the individual level. And then, as I said to you, maybe we thought what is left is a critical bottleneck, maybe in the backward connectivity from frontoparietal to specialized cortices. And then it's kind of nice, there is this interesting property that backward connectivity seems to be involved in long latency components. So maybe we can check also in patients if the long latency component can differentiate different levels of consciousness, right? So that's what we did, using now, again, like evoked potential, switching from fMRI to EEG, because you have a better temporal resolution to look at these forward versus backward processes, right? And so here it's a mismatch negativity with tones of different frequencies. So it's, for example, TD, TD, and the first tone is a deviant. So when you change the frequency, the first time you present a tone of a different frequency, it's the deviant. And then when you repeat it several times, it becomes a standard. But that paradigm is nice because at the end of the day, you compare deviant and standard that have the same physical properties. And so it's kind of nicely matched between conditions. And then here's the kind of signal you record at one electrode, and you see there is a difference in potential there. And then it has been very much studied with the kind of architecture I showed you before. You need some kind of network with auditory cortices, but also frontal cortex to generate that signal, and frontal and backward connections. So with these kind of tools, now we go to patients and see what's happening. And so the first thing is to look at the scalp level. What I was saying to you before is there relationship between long latency component amplitude and the level of consciousness, and it seems that there is. So here you have like the scalp topography, and here the propagation in time. In healthy volunteers, you see that there is widespread response, and then it's going until 400 milliseconds. In vegetative state, you find nothing after 100 milliseconds, and it's very localized. Minimally conscious state patients show a response that is going again until 400 milliseconds too. And when you look for an effect of the level of consciousness, you will see indeed that it's in the amplitude of the long latency connections that you can find that. So it's kind of promising. You see that indeed there seems to be a link between, in this paradigm, between the long latency components and the level of consciousness. But then what we really wanted to see is what's happening at the network level, right? So quantify the connectivity in the different connections of the network and see if there is a difference between controls and patients. And what we found is actually that where you can find a difference is not in the lower level cortices, but it's indeed, as we thought, in the backward connectivity between frontal cortex and the lower level cortices. So if you want to, so this is preserved in controls and in minimally conscious state, but this is impaired in patients in vegetative state. If you want to rephrase it differently, what we think is actually that even in patients in vegetative state, you have associative cortices that are involved, and then it's going, and at the group level, actually we see also that in patients in vegetative state, frontal cortex is involved. But what seems to be impaired is the backward connection from the frontal to the temporal cortices. And actually, we've done another recent study in anesthesia, using, again, dynamic causal modeling, which is also in favor of this specific preferential role of backward connectivity and the level of consciousness. Here is two areas from the default network. So you have the medial prefrontal cortex and precuneus. And we looked at connectivity during actually resting state EEG here, during propofol anesthesia versus normal wakefulness. And when you look at what kind of network can generate the EEG signal for these conditions, you see that actually when you compare, this is a comparison compared so this is connectivity in the network as compared to wakefulness. And you see that for sedation, when people are still conscious, you don't find a difference. But the backward connective from frontal to parietal cortex is impaired when you lose consciousness. So this is quite fitting well together, saying there might be a link between backward connectivity function and consciousness. So what I showed you, if you want to summarize now, is that when you look in coma or anesthesia or sleep, there seems to be a good correlation between the strength of connectivity, being functional or effective connectivity, in frontoparietal cortices and the level of consciousness. And this is true for functional MRI, for TMS-EEG. And then if you look at response to stimuli, we can also maybe think that there is a preferential role, a preferential importance of backward connections in the network. So you could think, oh, cool, now it's solved, yeah? Consciousness is linked to connectivity, and when connectivity is decreased, you're less conscious, and when connectivity is preserved, you're conscious. But actually, as I said, we want a general mechanism for the different states like anesthesia, coma, epilepsy, we want to find something that is true in different states so that we can come back to patients with that criteria, because patients are quite different from the other states. So then you want to look not only in coma, anesthesia, and sleep, but also in the other conditions. And this is quite informative. So, okay, that's what I said before. If you look in coma, vegetative state, anesthesia, you have a good correlation. When connectivity decreases, consciousness decreases. And you can say, so it's maybe when you're less conscious, your brain functions less. But think about epilepsy. Actually, in some cases, like generalized tonic-clonic seizures, when you measure brain metabolism, you have actually an increase in metabolism, not a decrease, an increase in frontoparietal cortex, exactly the same than before. Okay? So there is an increase in metabolism. And when you look at connectivity, as far as we can know, the evidence is saying for now that you have some cases where when you lose consciousness, like here is a comparison between seizures where you don't lose consciousness, that are in white, and seizures where you lose consciousness, and you look at connectivity between different regions in intracranial recordings, and you see that actually when you lose consciousness, there's increased connectivity everywhere between thalamus and precuneus, because precuneus and temporal cortex is kind of everywhere in the network. So then you cannot really say anymore that decreased connectivity equals decreased consciousness as a general rule, because sometimes you can have increased connectivity and then you lose consciousness anyway. And then the hypothesis is maybe you need something else than connectivity. Maybe in these kind of cases where in epileptic seizures, like the connectivity is everywhere, then you have a loss of differentiation of brain activity. Everybody's just doing the same job, right? So that's kind of the working hypothesis for now. But there is a lot of work needed in epilepsy to check that, right? But then we can still look again in coma and anesthesia and sleep, and actually we can see this loss of brain activity differentiation in the previous studies we've done too. So if you look, for example, here is in TMS-EEG. TMS-EEG is perfect for that. You have really like a sharp spike, and then you look at how the brain activity functions. This is an example of a patient in vegetative state. So stimulating in frontal or parietal regions, and you see actually here the source reconstructed response, okay? So in this patient, you see like a very local response, but then also when you look at the traces of what the regions are doing, everybody's doing the same job, okay? It's like the same phenomenon everywhere in the brain. When the patient transitions to minimally conscious state, this is a very different story. So not only the response spreads much more, but also now there is a recovery also of the differentiation of brain activity. Like you see, all the areas are doing something different now. And it's of course the case when they emerge from a minimally conscious state. There's like more connectivity, but also very strikingly, this is like very, very differentiated and complex. And this is like one single phenomenon, okay? So there seems to be in vegetative state too, and it's not the only patient, we have many more like that, a loss of differentiation of brain activity on top of the loss of connectivity. This is another example in anesthesia, using another property of TMS-EEG responses, which is that when you stimulate in the back of the brain, it's kind of as if you were resetting all the local oscillators. You actually uncover the natural frequency of the area. So when you stimulate in occipital cortex, typically you will have like a beautiful alpha response. And then when you stimulate in parietal cortex, they have like a Journal of Neuroscience paper showing that. When you stimulate in parietal, you will have some beta response. And in frontal, you have some like gamma response. And here is just an example of global mean field power, but you see indeed that in normal wakefulness, when you stimulate there, it's slower, and then it's going faster and faster, especially in that kind of shortly after the stimulation, okay? And in anesthesia, this is completely lost. So in propofol, whatever you stimulate, it will be very localized, but also it will be the same shape of the response. It's like very stereotypical. So this is examples where you have decreased connectivity in response to stimulation and also decrease of differentiation. But there are also some other studies, in particular with intracranial stimulation or in animals, where actually when you compare the strengths, it's a bit the same story than before. If you compare the strength of the response in anesthesia compared to wakefulness, you will have a stronger response to stimulation. So in our criteria, you will have stronger effective connectivity in this case. But the difference is, again, that even if the signal is propagating a lot in the brain, in anesthesia, everybody is doing the same job. While in wakefulness, so here is like Yeah, so you see here, this is parietal, occipital, and frontal, the three different traces of the response of the areas. In wakefulness, everybody's doing something different. In anesthesia, it's very stereotypical, and you have a lot of differentiation. Yeah, so actually, this is also showing you that even for response to external stimuli, or to TMS-EEG, if you really want to be specific on what is differentiation, like more conservative and reliable on what is differentiating a response during unconsciousness with a response during normal consciousness, you need not only the extent of propagation of stimuli, but also really to look at the differentiation of brain activity. So both are necessary. And yeah, just to mention, maybe we think that in sleep anesthesia, why we have that kind of stereotypical behavior is due to the presence of bistable dynamics, some kind of all-on response of the neurons, and that is marked in the EEG by the presence of slow waves, for example. And we did some studies showing that indeed in propofol and in sleep, it's a very similar thing. You have slow waves that really behave in a lot of ways in a very similar manner. In coma, it's not so known, but there are some indications that it might be also some likely phenomenon in a lot of patients. For example, if you look at the bispectral index, which is also looking a lot at the delta power in the EEG, there seems to be like a correlation with the level of consciousness in patients, but it's not really known. But we think that maybe the explanation for this kind of stereotypical behavior during unconsciousness in case of sleep and anesthesia is likely the presence of bistable dynamics. So I come to the conclusion now. Yeah. So if you want to summarize what I said before, I hope I could show you that you can find two different things, one or the other, when you lose consciousness. The first thing is you have a good correlation, usually between the level of consciousness and the strength of brain connectivity. So if brain connectivity decreases too much, then it's likely your brain won't be able to generate conscious perception. At the same time, connectivity can be preserved. So, and then you cannot say if the subject is conscious or not. You need something more. You need at least to check for the differentiation of brain activity. And then if you have these two criteria together, again, our working hypothesis would be that you have reasonable chances then to be able to differentiate brain activity during consciousness or in unconscious conditions. So, yeah, so how to go forward now? I mean, Stephen will talk better than me about that. But what we kind of need to do is to compare the different techniques together and to obtain some normative data. So I showed you like functional connectivity, TMS-EEG, external stimuli, all show some kind of correlation between, you can find some markers of the level of consciousness in all the techniques. We have to check at the individual patient level which one is a better predictor of the clinical state and also like of the prognosis. And then kind of also trying to better understand what are the precise mechanisms. Combining different techniques is really nice for that. And well, the modeling approach like dynamic causal modeling or computational modeling in general help us also understand, because we know more and more about general principles of brain function. So if we can use all that knowledge to better understand what's happening also in the lesion brain, it can be very useful. As I said, there's still some work to be done to really have a better characterization of the neural correlates of consciousness in the different states. So not only in coma vegetative state, also in anesthesia, sleep. But there is a lot lacking actually. Somnambulism or epilepsy, we don't know so much. And we need to know more if we want to really find some general principles of that. And at the end of the day, when you do that, you kind of take a step back from the data. And this kind of idea of finding general principles of mechanism leading to conscious perception is kind of taking a theoretical approach already. So there is a lot of interest actually from my side and other people to try to think about what could be the mechanism in the data and build some coherent theoretical approach. But the thing is, for now, all theories of consciousness, they are really, really at the beginning. And we need to be more precise and to bridge them to the brain and try to generate some predictions that we can test and go further like that to be able to maybe help us build a coherent diagnostic framework. And if you do that, actually, you kind of switch from like just checking the brain activity and see what is working, what we call exploratory correlates of consciousness to more explanatory ones. Try to find some correlates which are actually linked to the mechanism by which the brain is generating perception. So that's kind of the hope that one day we'll be able to do that. And I'd just like to finish by saying this. Maybe there is a reason why these frontoparietal cortices, we find them all the time more impaired when patients are unconscious, being in sleep anesthesia. Even in epilepsy, if you have an increase in metabolism, you have some kind of abnormal thing going on there, but it's always in frontoparietal cortices. And maybe the reason is that you have some evidence now that there's a hierarchy of brain connectivity. Some areas in the brain are more central or more connected than others. Like here, it's a functional resting state functional MRI study in healthy volunteers, or here, using anatomy, diffusion tensor imaging, you will find that these frontoparietal regions, exactly the same that we find impaired in consciousness, are actually the highly connected hubs of brain connectivity. It kind of makes sense that if you impair the function of big hubs of brain connectivity, you will have massive consequences on brain function in general, and especially some functions that likely require some integrated behavior of the whole brain, such as generating consciousness. So that's it, and of course, like the studies I showed are the results of a massive teamwork, especially the team in Liège, wonderful team in Liège, and our collaborators in the world. I want to thank Stephen also for his support, his long-term support, and I want to thank you for your attention.
We'll stay here. We'll just take a few questions. Thank you, Melanie. We have time for a few questions. Let me ask the first one. I've always been confused what is meant by cerebral integration. At the cellular level, dendrites and cell body receive and integrate inputs to an output for firing. But at the cerebral level, are you referring to the back to front activity, or is something else meant by that?
Well, I think likely both forward and backward are important. And then it's kind of not really clear at what spatial scale you should look for consciousness, right? Right. At the same time, in the brain imaging approach, we take the kind of approach where we think maybe looking at the system at a really global, large-scale level is relevant. And we find some correlates at that scale. But I agree with you, there is no real ground truth or consensus at what scale you should look now.
But what do they mean by integration, cerebral integration?
Well, what I mean in these kind of studies is what we can measure by connectivity, being causal interactions, and if you can, between brain areas or parts of the brain, and kind of if you can measure them with functional imaging, the spatial scale you're looking at is actually the population level, more than the single neuron level. This doesn't mean this is a ground truth, but actually looking at the population level, we have a good correlation between causal interactions and consciousness.
Over there.
Hi. Some really nice takes on it, but there seems to be three aspects to whenever we do these kind of studies. And one is the idea of consciousness, the generation of the experience. Two is the ability to report, and three is the ability to remember, so memory consolidation. So the ability to report you talked about right at the beginning, and there's obviously locked-in syndrome, and people are unable, and now I think a lot of people, excuse the pun, are waking up to the idea that we have to sort of consider this, and this is a terrible thing when people are locked in. Clinicians, often were dismissive, I think. But the third one is actually the memory consolidation is, I think, less considered, and it may undermine the entire field. And what I mean by that is, imagine that these sort of other parts of the brain, which are definitely lighting up in your studies, maybe less so, but you've admitted that you can't correlate necessarily activity to consciousness, epilepsy being the paradigm case, maybe. So what if these other parts of the brain are conscious, fully experiential, but we're not consolidating memories? So there's the possibility of a terrible, maybe it's not as terrible as remembering, but since we're basing the whole science on the ability to report, or the person who's awake, what's happening if people are undergoing surgery or under conditions where these other parts of the brain are experiencing pain, maybe are other things that are fully conscious, but we have no way to access them. How do we begin to even consider that or deal with that possibility?
So the question about memory versus consciousness is actually a difficult one. And it's not only in coma, it's also in anesthesia. I'm sure Jean is even more suited than me to answer to that question. I would say from my point of view, it's unlikely it's only memory that would be impaired in these patients because, again, we see that widespread frontoparietal network dysfunction in them. And in patients who have only an impairment in memory, like normal subject amnestic patients, actually most people still consider them as conscious, so you don't need memory for consciousness neither. But still, it's a very difficult question.
Right,
because indeed, we're like largely based on report, and same question sometimes people ask during sleep. Okay, if you don't, because we say consciousness is what disappears during dreamless sleep. But then when you wake up, do you don't remember or not? Actually, we take the assumption that first, as a clinician, you have any way to study and to find something. You know, you have like to try to find a solution. Second is, from my point of view, I think it's not only that is happening in these kind of situations.
As an anesthesiologist, let me just say we're faced with that question all the time. How do we know our patients are not awake, experience, and just don't remember? And because we can't prove consciousness one way or the other, we can't rule it out. However, as George will tell us, when patients get light under anesthesia, there's all kinds of arousal and autonomic signs that we can detect. So I can say pretty certainly that our patients are not generally awake and just don't remember. One more question over there, then we got to move on.
As a general comment, this is a great talk. It's a kind of a tour de force, so much so that I'd like to hear it again in slow motion. Are your slides going to be available after this, or do you have a particular review paper that you think summarizes all this very well?
You can give me your email, I can send them to you, for example. Or we'll see, we can put them on the Coma Science Group website. All the papers we have are actually on Stephen's website. But I can also give you the slides if you want. Yeah.
All right, thank you.
One more quick one over here.
Yeah, I have a very short question since we were talking about the precise definition of terms that we use, and Stu had a question about cerebral integration. What do you understand when you say connectivity? Are you actually suggesting that those networks are directly connected neurons? If you show a blockbuster, then suddenly all the TVs show up. They are not connected whatsoever.
So there are different point of views from people on that question. And it's, again, as I said, there is no definite answer to that. There are some, for example, if you talk to Carl Friston about that kind of scale, he would say that likely the relevant scale for brain integration for us to look at for cognitive neuroscience would be the population level, and that maybe the individual neurons are too small to have really an effect on whole brain function, and could be considered as some kind of a noise that could be useful in many different ways, but maybe individual neurons would be too little to really have relevance. But that's, again, a point of view. I kind of sometimes have a tendency to agree with him, but I'm still not sure neither. Though, again, as a clinician, if you want to find a marker of consciousness, it seems that functional neuroimaging can provide a lot of information. But that is a correlation, not a causation, and no. You know, like, there is a very long discussion there. Okay?
Would gap junctions count as connections, connectivity? Gap junction connections?
Oh, yeah, certainly. But here we cannot really say.
You can't rule out one type chemical.
I don't think with this data you cannot rule out anything.
Let's thank Melanie for a really wonderful talk.